Hello, biology students, and welcome to Chapter 12. In this chapter, we're going to learn all about the chromosomal basis of inheritance. This is a very integrative chapter. It brings in what we've learned in mitosis and meiosis, and it also brings in the information that we learned in the chapter on Mendelian genetics. So let's go ahead and get started. The first of my videos in Chapter 12 is going to focus on chromosomes and sex linkage. Now Mendel, he knew a lot of stuff, right? He figured out just by looking at the phenotype of species and doing very careful crosses, he was able to figure out how traits were inherited. But he did not know that these hereditary factors that he talked about, these alleles, these genes, he did not know that they were located on chromosomes. And as far as he could figure out, everything was inherited kind of uniquely and separately. But we now know that genes are located on chromosomes, and that's a very important thing to think about. You can see a couple of genes have been fluorescently tagged here. So this chromosome and this chromosome have the same genes on them. That means that they're homologous chromosomes, right? Kind of tying things back into our cell division chapter. So mitosis and meiosis were first described in the late 1800s. And that's when we started coming up with this idea of the chromosomal theory of inheritance. And so there's a couple of key things here with the chromosome theory of inheritance. Number one, Mendel's genes have specific loci, and that's loci is plural for locus, and that just means position or location on chromosomes. And then, of course, chromosomes undergo segregation and independent assortment. So, like I said, we're going to be bringing things together, and that's the very important concepts right here, especially the second one about segregation and independent assortment, because those are Mendel's laws. We're going to apply them to chromosomes because chromosomes are the things that hold the genes. So let's take a look at meiosis again, and we're overlapping on this figure specific genes. We've got the Y gene for yellow versus green, and we have the R gene for round versus wrinkled. And in the first generation, this parental generation right here, remember the P generation, everything is true breeding. So big Y, and big R crosses with little y, and little r. And of course, when you're making gametes from something that's true breeding, there's really only the one kind of gamete that you can make, and that's you're segregating, right? And you're making them haploid. So this one has a big R and a big Y in it, and this one has a little r and a little y. If you breed these two homozygous individuals together, you're going to get what Mendel called the F1 generation. And in the F1 generation, your individuals, again, are all heterozygous. When individuals are heterozygous, we really have to start paying attention to Mendel's two laws, the law of segregation and the law of independent assortment. Now, don't get me wrong. It was happening, both of those laws were happening up here in the parental generation. But when it comes to the F1 generation, we really can see what's going on here with each of those two laws. Remember that the law of segregation says that when you're making gametes, you have to put the two alleles for each gene into separate gametes during gamete formation. So the big R is going to segregate from the little r. And you can see that lining up right here. This would be the metaphase plate right here in the center of this cell, and big R would be going towards one pole, and little r would be going towards the other pole. Same thing for Ys. Big Y would be going towards one pole, and little y would be going towards the other. And that's segregation. But when we have heterozygotes, we really have to also be paying attention to the law of independent assortment. What this law says is that when you've got alleles on, and this is important, on non-homologous chromosomes, right? So this chromosome right here that has the R gene is not homologous to the chromosome that carries the Y gene. So we could say these are non-homologous chromosomes. Just as a refresher, these two would be homologous. The one with the big R and the one with the little r would be homologous to each other. So the law of independent assortment says that when you have alleles on non-homologous chromosomes, that when you're going through gamete formation, you could basically arrange them just about any way. Like it could be that the little r is on this kind of heading towards the same pole as the little y, and the big R is kind of heading towards the same pole as the big Y. But that's equally as probable as this setup over here, where the big R and the little y are going to be going towards their own cell, and the big Y and the little r are heading towards their own cell. So this idea of independent assortment, it says basically there's two equally probable ways of lining up during the metaphase plate of metaphase 1. And this is only if you have 2 chromosomes. If you add more chromosomes, then you're adding more complexity and there's more options. But in the end here, we end up with 4 distinct kind of combinations of gametes in the end. And some of them are repeats, remember, because you have to separate your sister chromatids from each other. So Thomas Hunt Morgan did an experiment in the early 1900s. That's the 20th century, the last century, and he was looking at fruit flies, and he picked fruit flies for very similar reasons that that Mendel picked pea plants, because they produce lots of offspring, they can breed fairly quickly. In the case of fruit flies, every 2 weeks you have a new generation. And importantly, they only have 4 pairs of chromosomes. If we look up here in this example, there was 2 pairs of chromosomes. So 4 pairs of chromosomes is pretty good. It makes things fairly easy. Unlike us, where we have 23 pairs of chromosomes, things can get pretty complicated pretty quickly. So he was looking at eye color, and he noticed there was something called the wild type, or the normal phenotype. It's the most common phenotype, and that is red eyes. Anything that wasn't the wild type, those were referred to as mutant phenotypes. So in this case, you see a mutant phenotype of white eyes. The first mutant phenotype was white eyes, and so that's why we call that the mutant type. So when we're looking at his experiments, he also did what Mendel did, where he had a parental generation, an F1 generation, and an F2 generation. And very interestingly, and notice right here, this is going to become important in a second, that here's your red eye fly, and this is a true breeder because it's the parental generation, right? This one happens to be a female, and this is important. This was a white-eyed male, and when you cross the red-eyed female and the white-eyed male, you have offspring that are all red. What you can tell from this is that red is dominant. So not only is red wild type, which is the most common, it's also dominant. On the other hand, if you look at the F2 generation, you can see, there's that 3-to-1 ratio that we were absolutely expecting from across, with the dominant being red and recessive being white. But here's really important, only males had white eyes. This is a huge deal. It's like a mind-blowing idea that how could it be that only males had white eyes? And so what Morgan concluded is that eye color is related to the sex of the fly. And so he figured out that the way this would work is if the gene was located on the X chromosome. So fruit flies do determine sex very similar to the way we do with X chromosomes and Y chromosomes. And if you look at the symbols here, you'll notice the X chromosome and Y chromosome here. But the name of the alleles is a little different, and I wanted to point this out. So Morgan used the term W plus, and plus was referring to the wild type. So W plus means the wild type, and just plain W meant the mutant type. And notice that we're not using bigs and littles in this case because Morgan used a different system than Mendel did, and the idea of wild type was important to him. But we can still figure out dominance and recessiveness based on the way that that the F1 generation looked. So if we segregate these alleles, and we have repeats, so no need to write down repeats, right? Things are easier if you don't write down repeats. All of the alleles that this female can make are the W plus or the wild type bred. And the male has 2 different chromosomes, so he can actually, we do have to segregate those chromosomes from each other. So we're going to put the mutant white allele in one gamete, and the mutant, or sorry, the Y is not, chromosome's not mutant, but the Y chromosome into the other. Please notice that the Y chromosome does not have the allele on it, because the allele is found on the X chromosome, not the Y chromosome. So here's our female gamete. Again, we could have written the repeat, but we didn't. And if you combine this egg and the sperm, you have the wild type and the mutant type, but they're showing red. And it's female because 2 X chromosomes. The male also has red eyes because he inherited that wild type red from his mother and the Y chromosome from his father, and he's only going to be able to show the wild type red eye. When these 2 mate with each other, we really start to see the interesting form of inheritance. So the wild type, the male sperm is either wild type X chromosome or Y chromosome. And yes, you really do have to put the Y chromosome there, because if you don't put the Y chromosome there, then you're not going to make any male babies, right? And then the eggs, they're heterozygous. So here's the wild type red and the mutant white. And when you fill in the Punnett square, what you see is a 3-to-1 ratio. But it's not really a 3-to-1 ratio. It's a 2-to-1-to-1 ratio. So I'm going to now write down that 2-to-1-to-1 ratio. It's 2 red-eyed females to 1 red-eyed male to 1 white-eyed male. Hope that makes sense. When you're doing sex-linked traits, you have to say the gender or sex of the individual along with whatever trait that you're looking at. So humans and other mammals, as well as fruit flies apparently, they have 2 types of sex chromosome, the larger X chromosome and the smaller Y chromosome. Individuals who inherit 2 X chromosomes develop anatomy that we associate with a female sex. And individuals that inherit a Y chromosome, as well as the X chromosome, are considered to be male. And so that's one of the reasons we tend to look at chromosomes with designating male versus female. But I mean, I just want to put it out there that doesn't mean that that's how you identify. There's a lot of fluidity in this, and it's not a deterministic thing. It does, in fact, we know lots of exceptions to, for example, that certain regions of the Y chromosome can cross over, and we'll talk about this a little bit later, but mutations can happen where parts of the X chromosome are found on the Y chromosome. And so there's all sorts of things that go along with sex determination besides the simple XY thing. Just kind of heads up on that. So, but if we just look at the basics here, only the very ends of the Y chromosome have regions that are homologous with the regions of the X chromosome. This allows the X and Y chromosome to pair up during meiosis, to become tetrads during meiosis. And And of course, that would only happen in males because males are the only ones that have the Y chromosome. In females, of course, X chromosomes are completely homologous to other X chromosomes. So we don't have to be so concerned about that, how they line up. Just kind of think about the way we're going to count chromosomes. We know that humans have a total of 46 chromosomes, right? 46 chromosomes. But we can break them down into our autosomes, which are these 44 chromosomes, and the sex chromosomes, which are these 2 chromosomes. Other animals do have other ways of determining sex, just so that you know. And of course, plants don't have, most plants anyway, don't have sex associated with them. They have both male and female parts. But there are other different ways of doing this. Birds determine sex a little differently. Other insects besides fruit flies do, like butterflies, determine sex differently from the way that humans and other mammals determine sex. So just kind of heads up. So if a gene is located on either sex chromosome, either the X chromosome or the Y chromosome, we say it's a sex-linked gene. If the gene is found on the X chromosome alone, we call that an X-linked gene. And if genes are located on the Y chromosome, clearly we're going to be calling that a Y-linked gene. But there are very few Y-linked genes. So for the most part, a lot of times we use the word sex-linked and X-linked interchangeably. Again, that's just because for the most part, there's not a lot of genes on the Y chromosome. Many Y-linked genes, though, do help determine sex. In fact, there's this region of the Y chromosome called the sex determination region, or the SRY region of the Y chromosome. And there's this other region called the AZF that's important in sperm development. But if we look at the X chromosome, there are a lot of genes on there that are unrelated to sex, including ocular albinism, that's where your eyes don't have any melanin, Duchenne's muscular dystrophy, there's a region that's important in androgen insensitivity, also severe combined immunodeficiency syndrome, hemophilia, and colorblindness are all genes that are located on the X chromosome. And so they're going to be inherited differently from how we've been talking about with Mendel. One of the ways that we show sex-linked or X-linked crosses is by when you're deciding on allele names, you need to use the letter X and Y and then say, oh, this X chromosome has a trait on it. And again, the reason for this is because females, right? Females have 2 X chromosomes, and males only have 1 X chromosome, and their other chromosome is the Y chromosome. If you don't say that you've got the traits are on the chromosomes, then you do encounter some problems when you're doing when you're doing Punnett squares. So it is very possible for females to express X-linked traits, but if that's true, then the females have to have 2 copies if it's a recessive allele, they have to have 2 copies. So this female right here that I'm showing you, she's homozygous dominant. This male right here, well, we can't even use the word homozygous because he doesn't have 2 of the same. We actually use the word hemizygous to refer to the fact that he's showing recessive, but he only has the 1 allele. So if you're going to have a female that's showing a recessive allele, she has to be homozygous recessive. And so that's less common than showing just one of them. X-linked, what I'm trying to say with this is that X-linked recessive disorders are much more common in males than females because males only have to have 1 copy of that allele, and females have to have 2 copies of that allele. So in this problem right here, we've got a colorblind father and a mother with normal vision, and notice that all the children here have normal vision. In this case right here, where the mother's heterozygous and the father has normal vision, if you look at that, we can actually see how a male with color blindness could be conceived. In fact, for males, there's a 50% chance that they will be colorblind, but there's a 0% chance that the female will be colorblind. In this case, and this is a bit of a misprint here. That should be a big N. In this case, we have a female being homozygous for not colorblind, but also right here we see that she's a carrier. If you have a female who's a carrier and a male who's colorblind, you can have a female who is colorblind right there. And you can also, of course, have that male who's colorblind. So there's a 1 to 1 to 1 to 1 ratio of colorblindness and sex in this particular cross. I already listed some of these before, but they're ones that do come up over and over again in genetics problems. So just kind of listing them for you here, that colorblindness is mostly X-linked. We just tend to simplify it and talk about it being X-linked. Duchenne muscular dystrophy and hemophilia, those often tend to show up in genetics problems. So just kind of heads up. Now, when we deal with X chromosomes, there's something else interesting that happens in mammals with the X chromosome. And that is that, females have 2 X chromosomes in each cell. But I know this sounds strange, but one of those X chromosomes is inactivated. We call it X inactivation in female mammals. It's inactivated during embryonic development. So we call this a Barr body. If the X chromosome becomes inactive, it condenses, it's always coiled up into this structure called a Barr body. If a female is heterozygous for a particular gene located on the X chromosome, she will have a mosaic for that character. In other words, she might not be completely colorblind. Maybe she'll be colorblind in one eye, but not in the other eye. Or something that's interesting in cats, which a lot of people are very interested in, is this, it's just so very, very physical. You can see this, that the color of cats is located on the X chromosome. And so there's an allele for orange fur in this case, and an allele for black fur, but they're on the X chromosome. And so when we go through the developmental process, when cats go through their developmental process, some of them will inactivate the orange allele, the orange chromosome of their 2 X chromosomes. And some of the cells will inactivate the black chromosome, the one with the black allele on it. And the active one will be the one with the orange. And so you get this appearance called tortoise shell, where some of the cat is black and some of the cat is orange. So there's 2 separate cell populations in this cat, black and orange. I think that's really cool, and it's a good introduction to the concept of Barr bodies. Well, that's all I have for you for part 1 of chapter 12. I'll be picking this up again in part 2, where we talked about linked genes and their effect on crossing over.